So my name is Dick Vethaak and I work as a water quality specialist at Deltares, which is in Delft, the Netherlands. And I'm also a professor of ecotoxicology at the University of Amsterdam. I first came aware of this, of the microplastic or the plastic pollution problem, I think it was in 2008 or 2009 when, well, I think it was the time when all these pictures of birds that were, full of plastics and they died. Because of suffocation of the plastics. And then I started my own research line on this subject, particularly looking at the fate and effects of plastics, not just only in the marine environment, but also on the freshwater side, in the freshwater environment. So to see how these plastics, where are the sources, how can we tackle the sources, how can we in fact stop this increasing serious environmental problem. So I'm particularly enthusiastic about teaching in terms of PhD students and masters, but I also like to do fundamental research and applied research. So I'm a bit of everything, but you can find me a lot in the field. I like the field affinity, to see what's happening at sea is for me important. I need to see the trash and the pollution to make sense. But at the same time, I'm writing papers, of course, and I'm doing research in the lab. So that's me. So what do we know so far about microplastics? Well, what we know, they are widespread in the marine environment. You will find them everywhere in oceans, from the rivers to the deep sea, and even in the Arctic ice and also atmosphere. Currently, there are clear limitations in analytical methods. This means that we can only really accurately measure environmental microplastics in the larger micro-size range. The detection level of microplastics in field samples is about 10 to 100 micrometer. And however, it's very likely there are smaller size particles present in the environment, including those in the nano-size range. We're not able to measure them or detect them. So this is a clear limitation. And then another point is, of course, that the marine organisms are exposed to microplastics via the same pathways used to food, including filtration, active grazing and deposit feeding, and also ventilation via transport across the gills. And the last point I would like to mention is that we know that microplastics are ingested by a wide range of marine organisms, including invertebrates, fish, birds and marine mammals. And in some of these organisms, the incidence of ingestion is widespread across populations. So, having said that, then the question arises, so what are the biological impacts of microplastics? And I have, on this slide here, I have summarized some of the potential adverse effects of microplastics on marine organisms. And you can classify them in a couple of different groups here. There are the physical effects, which are the obstruction or damage of feeding organs or digestive tract or other physical harm. And then there's a second group, the particle toxicity, which is in fact when the particles are taken up and translocated into tissues, cells and body fluids, they can cause cellular toxicity and also pathological changes. We call that particle toxicity. The third group is chemical toxicity. Microplastic can act as factors for chemical transport into organisms. And then we have a fourth group here, which are the environmental factor effects. Microplastics as carrier for the dispersal of chemicals and biota. And biota in this case you can think of invasive species, but also pathogenic microorganisms. And finally, there is, of course, the group of the higher level effects, and then you can think of population, community, and ecosystem effects. So we probably all are familiar with these sort of pictures where, marine mammals eat or become entangled in plastic pollution. But what about the microplastics? We know that the likelihood of ingestion will increase with decreasing particle size. So then the question arises, do ingested microplastic present a mechanical hazard to small animals like plankton or worms or bivalves, similar to those effects observed for microplastic in larger animals? And so far, there are only few field studies on larger sizes of microplastics in birds and fish, so there are no records for entanglement or obstruction of feeding organs, where we look at the more smaller particles and the smaller animals. And there are also no clear indication of adverse effects of mechanical obstruction. For example, no clear relation was found between the condition factor of North Sea fish and the presence of ingested microplastics. Now, well, in contrast to the field situation, effects of microplastics have been reported in numerous lab studies with a wide variety of marine species exposed to different microplastics. And I have summarized in this table some of the examples. In the example report you will find many more of them, but just to illustrate some of the effects observed in lab exposure studies. For example, one study found that absorption of nanoplastics to algae hindered algae photosynthesis and appeared to induce oxidative stress. Another example here, this is about polystyrene microplastic adhered to the external feeding organs of exposed zooplankton and showed reduced algal feeding and also reduced reproductive output. There are several other cases. For example, the case of the mussels, microplastics showed reduced filtering and feeding activity after exposure to polystyrene and polyethylene. And finally, fish exposed to nanoplastics showing altered behavior and also altered fat metabolisms. If you look at these studies, these lab exposures, there are a couple of points that you can conclude from that. So the observed effects are often related to the reduction in energy reserves. That is the first point. Secondly, exposure mostly in lower micro-nano range, meaning that all these lab experiments, most of them are done with the smaller sized microplastics and preferably the nanoplastics. And then the third point is that short-term and high exposure levels. These experiments, they use very high exposure levels that are not really comparable to the field situation, so they have in general a low field relevance. So perhaps a more environmentally realistic study is the one with lugworms exposed to PVC particles from Stephanie Wright and co-authors. And in this case, the lugworms were exposed to so-called spiked sediments. They spiked the sediments with PVC particles that were, the sizes were similar to those of the sediments. So let's say a more environmental realistic situation. And the authors found statistically significant effects on the organism's fitness and bioaccumulation. 1% PVC spiked in the sediment significantly decreased energy reserve by 30%, and a 5% PVC level significantly decreased energy reserve by 50%. These exposure levels, let's say the 1%, are used in this experiment are close to the one we can encounter in hotspots. But I would say this is an environmental realistic example. Lugworms, like many marine organisms, have the ability to remove unwanted natural particulates from the body without causing harm. However, once ingested, there is the potential for microplastics to be absorbed into the body via translocation across epithelial membranes. And it's well known that internalized particles may evoke an immune response. We can also call that foreign body reaction with granuloma formation. And this is what we call particle toxicity. And there are a few studies of marine organisms that have clearly demonstrated such direct particle toxicity effects of microplastics. And a good example is the study of Nadia von Moss and co-authors. And they exposed mussels to primary high-density polyethylene plastic powders in the size of 0 to, let's say, almost 0 to 80 micrometer, so smaller than 80 micrometer, but also including the nano-sized particles. And they found that it was absorbed by the digestive gland vacuoles and taken up in the hemolymph. And they also found a strong immune response after 96 hours in the digestive gland and the hemolymph. So the adverse effects of toxicity they showed includes oxidative stress, a lower lysomal membrane stability activity, and also all sorts of histopathological changes, including inflammation and granuloma formation. And this is typical what we call particle toxicity. Particles were found also in the cell organelles, so not just only in the hemolymph. And this study provides proof of principle that microplastics are taken up into cells and cause significant effects on tissues and cells. So we already heard about the fact that microplastics can serve as a factor of hazardous chemicals, including POPs, to marine organisms. But in many cases, they are also exposed to the same chemicals through prey. Conclusive evidence of adverse effects caused by chemicals associated with microplastics is quite difficult to obtain because so far there are only few studies available that compare both pathways. So let's say the exposure via the food and the post-exposure via the plastics. What we know so far is from model calculations, and they suggest that these are studies by the group of Bart Koolmans and co-authors in the Netherlands, and they suggest that the magnitude of impact of microplastics on bioaccumulation of POPs is quite small. And also the role of microplastics in delivering POPs to higher trophic levels is minor. Now, having said that, there is some emerging evidence now that, well, for showing effects of plastic-derived chemicals in tissues and adverse effects. One is a field study on birds by Tanaka et al. And there's another lab study that demonstrates the transfer of POPs from plastic to fish and adverse effects from environmental relevant concentrations by Chelsea Rockman. And that later study showed that fish exposed to a mixture of polyethylene with chemical pollutants sorbed from the marine environment, they bioaccumulate these chemicals and they suffer from liver toxicity and pathology. And by contrast, so if you look at the fish fat, virgin, so to say, clean polyethylene fragments also show signs of stress, but less severe than fish fat marine plastic fragments. So again, here you see how that it's important to compare both pathways to come to any conclusion. A next category of potential impacts is the potential impacts or effects on populations, community and ecosystems. Well, in general, I can say that our knowledge indicating this sort of higher level effects caused by microplastics in the marine environment is really poor. But there are a couple of potential impacts that are recognized at the moment. First of all, microplastics may act as new habitats or new food sources for marine organisms. You can think of microbial communities. But a good example is the sea skater. It's a pelagic insect that uses the plastic to lay their eggs on. So there's a paper that shows a correlation between the amount of plastics and the population density of this insect. Marine debris, as also supposed for floating seaweeds, may also increase gene flow thus allowing genetic mixing among populations and decreasing genetic variability within populations. And there's another example here of a potential effect, and that's the increase in biofilm on plastics. Microplastics may increase the food availability to, for example, zooplankton. And then as such an effect may have a bottom-up effect on plankton communities. And I also showed you already, in this lab exposure experiments, that microplastics may have an effect on green algae, so phytoplankton, and also copepods, secondary producers. So, which may have a bottom-up effect, again, on plankton communities, but also on the productivity of the whole ecosystem. And a last example here of a potential effect is the biofilm formation on plastic that is linked to the productivity of surrounding seawater. So there are a couple of potential effects, but in general our knowledge is of higher level effects is really very poor. Particle toxicity is a well-known phenomenon in mammalian toxicology. And I have a table here that shows you some examples. Again, in the exam report you find many more of them. And as you can see here from this study here, you can see that when humans and rodents ingest microplastics smaller than 150 micrometer, they have been shown to translocate from the gut to the lymph and circulatory systems. Another study demonstrated how nano-sized plastic particles up to 250 nanometer in diameter are capable to cross the human placenta. And more knowledge of the transfer of microplastics through biological membranes can also be mined from the emerging field of nanomedicines. From this field we know that nanoplastics are used for targeted drug delivery and release, including the attention to toxicity for these particles. So in general, and you can see that also from the table again here, that there are studies that show that after translocation that adverse effects may start to emerge due to interactions of these particles with cells and tissues, and that in fact microparticles of plastic in the human body or mammals are unhealthy. So what we can learn from these mammalian exposure studies is first of all that the size is very important. Size matters in this case. Nano-sized particles have been shown to cross membranes under laboratory conditions and causing tissue damage. And there's evidence that microplastics in a human body or mammals are unhealthy. And human exposure is also a concern if seafood containing microplastics is consumed. The potential accumulation of microplastics in the food chain, especially in fish and shellfish, could have consequences for the health of human consumers. And there are a couple of studies that have investigated the ingestion of microplastic by several commercial species and especially shellfish from Europe. And these studies, they refer to the blue mussels from the Belgian-Dutch coast, and they show that a gram wet weight product of mussel can contain several microplastics. And if you express that on the basis of the annual dietary exposure for European shellfish consumers, then you can amount to 10,000 to 11,000 microplastics per year. What does that mean? Well, I don't know. Probably most of these microplastics you ingest will leave your body again. But the question, of course, is again, what about the very small particles that are capable of crossing the membranes and entering your body? So it's evident that humans are exposed to microplastics through their diet and the presence of microplastics in seafood, and that it could pose a threat to the food safety. But as you can see here from this slide, there's a pot of honey there, because microplastics have also recently been detected in all sorts of terrestrial products, like honey, sugar, milk. So a risk assessment should therefore comprise the dietary exposure from a range of foods across the total diet in order to assess the contributing risk of contaminated marine food items. So the attributable risk of marine foodstuff to the total diet. So what do we know about microplastic toxicity so far? There are early warnings. There are early warnings for plastic particle toxicity, like the case of the mussel, for example. That effects observed in lab studies often are at high exposure levels, But that the field exposure data is mostly for particles that are much larger. So let's say larger than 50 or 300 micrometer. So there's a sort of mismatch between the field and the lab situation. And as we call that, so the data from the lab exposure study, we call that hazard data is mostly for the nanoparticle sizes. And the field data is mostly for the micro sizes. Consequently, the human exposure hazard data is a dark black box. The risk assessment and the data are too limited to do a proper, adequate risk assessment to support risk-based decision making. And if you want to assess the risk, you need knowledge about the hazard and also the exposure. Risk is the product of hazard and exposure. So I have a take-home message here is that we have to learn from the field of ecotoxicology of engineered nanoparticles. I think we can learn a lot about that. And also from the field of mixture toxicity and risk assessment. Microplastics, in fact, are mixtures, complex mixtures of contaminants. And to tackle them, you need to apply the mixture toxicity and risk assessment concepts. So what we learned from all these studies I showed you and discussed with you is that the risk of associated effects following exposure to microplastics will depend on the following. First of all, the number of particles, which makes sense. Higher concentrations, higher exposure levels. But also the size distribution. Size really matters. The smaller sized particles are more hazardous than the larger microplastics. Shape is important. Surface properties. We know that the smaller particles have a higher volume to surface ratio and so a higher chemical reactivity. But also more surface, for example, for microorganisms to grow on. The polymer composition is important. Not all plastics are the same. They have different compositions. There are also copolymers and the density of the particles. And then there's the duration of exposure, which is important. And most of these lab studies now, they are short-term. And what we really need is the long-term experiments with the more, let's say, environmental realistic concentrations. Another point is the kinetics of absorption and desorption of contaminants with respect to the plastic and the organism. And then finally, the biology of the organism. The biology is very important. Are you a filter feeder? Are you a deposit feeder? So how do you feed? What is your feeding guild? Important also, we know that different species could have different gut environments, would play a role also in the availability and bioavailability of contaminants from plastics, for example. So all these sort of factors should be considered when you attempt to assess the risk of the associated effects following exposure to microplastics. So asking me about what are the most urgent research needs in the field of micro and nanoplastic, particularly when you look at the potential impacts of microplastics, then I have 4 areas that deserve special attention. First of all, chronic exposure experiments. I showed you before that most of the lab exposures are done short-term with very high concentrations. So we need really long-term experiments with relevant concentrations, environmental relevant concentrations. That is the first point. Second point refers to the whole problem of the nanoplastics, the nano-sized plastics and the nanoplastic. This is something, we don't even know if they occur in the environment. So first of all, we need to develop analytical methods, and then it's the question of what is the harm to marine organisms and also ultimately to us, to humans. So this is an important point. And we know that the nanoplastics are more hazardous than the microplastics for several reasons. They can cross biological membranes. That is one thing, and the other thing is they have a larger volume to surface ratio, so they have a higher chemical reactivity, so they can absorb more contaminants from the environment. And all these aspects make them more hazardous than the larger particles. So that, in fact, the combination of the two, the absorption of more contaminants together with the hazard that they can cross, the potential that they cross epithelium membranes, is what they call the Trojan horse principle. So they can bring these contaminants in your body, and they can transfer them to organs and distribute them to places where the contaminants maybe don't end up via the food. Think about the brain, or think about the placenta. So having said that, then there's a third area which we call the environmental factor of dispersal so the microplastics can be filled with organisms, and they can be dispersed because those microplastics, they float around the whole world, or via the air even. So they can contain exotic organisms, invasive species, but also microorganisms, bacteria, pathogens also. We have already an example of a virus that is distributed in that way and causing a serious hazard. So all these aspects are very important, but at the end, of course, the question is, what is it doing to us? So to human health. Is it posing a threat to us? So, and I think that is a very big black box that really deserves special attention. And I know there are a couple of projects starting to look at it now, but I think there must be a much bigger effort, to find out about that crucial question. What is the impact on us, human health?